So now we are here at the prediction modeling protein structure. This is also another really exciting area of bioinformatics. I know that our speaker, Dr. Kate Cooper, was talking about some really cool and amazing areas to go into in bioinformatics, and those were very, very cool. And I think this is like, this is, those areas that you start, like nutrition science and all those things, that those are actually very connected to this protein folding area, because we're actually looking to see how different types of proteins fold together. And so I think that really all research, I claim, I think all research in bioinformatics is really something to do with protein folding. I mean, it connects with protein folding in some way. And so when she was talking about like how different how the biome reacts to certain types of foods that you may be eating, those foods have they have interactions with the protein of your of the stuff in your biome, and how those interactions fare is more or less the kinds of health that you can expect to have. And so I'd say that folding is probably one of those big things. It's all over the place. But one thing we were talking about with folding, though, and I think this is one of my favorite graphics to use, is that if you have a piece of paper, you can fold it into a folded crane, which is a desk ornament. It won't fly, but it's a bird. Or you can take that piece of paper and fold it into a paper airplane, and it will fly. And so it's the same piece of material. It's just that how you fold it gives it this this structure that allows it to have certain types of capabilities. And so if you look at most of the stuff around you, everything has a certain type of structure about it, which allows you to function with this thing the way that it's the design gives you this the ability to have to interact with this in specific ways. Like, for instance, the design of your computer right now, for instance, like where your keyboard is, where the screen is. I mean, those things are actually done on purpose so that you can actually like rest your hands on your computer, and you can look over your hands and see your screen. And so these are all design features. And so proteins are exactly the same way. The way that a protein physically is kind of put together, like how it actually is designed, makes it into this. That's how it's able to do its perform its functions here. And so what we're seeing here is a three-dimensional view of a protein, and all these we have these curly bits and these the chords, the helices, the sheets. We'll talk about those in a second here, but all of those things really come out of the sequence, which is like right here. So this sequence is actually kind of like it's the amino acids, but when those amino acids are able to come into their own special, what they call stable state or protein conformation, they look like this. They're like springs, and they kind of like come together in a specific way. It's like, for instance, this is how the protein is in its relaxed and stable state, so it's not going to change. But that, I should say one thing though, and that is that particular way of looking at that protein is the stable state of that protein in this current environment. Which means that if you have a protein which is in a different environment, maybe it's hotter or it's colder, or maybe there's more salt in the environment, or maybe there's less salt, maybe there's glucose floating around, maybe there's some other toxin or chemical, or maybe this protein is floating in space where there's no gravity. These are all what they call environmental shocks. And so these environmental shocks actually do change the way that a protein is able to fold. And so you might find, for instance, that a protein looks like this when it's at room temperature, but when it's heated up, it becomes much more different. It looks much more different. And one of the things that we discussed about this whole thing was that if you take an egg, the egg yolk is protein, and at room temperature, it is liquid. But if you put it into boiling water and make a boiled egg, then you denature you're basically breaking the protein, because you're subjecting it to such a shock from heat shock, that it's been snapped into a different conformation, and that new conformation makes it. I mean, it's now impossible to go back to the original conformation because of such the changes that it sustained. And so what I'm talking about though is this protein folding stage is like the fourth stage in the central dogma of biology: DNA, RNA, protein. Well, when you get to the protein stage, like when you talk about the DNA, then the RNA, and then the protein, it seems to me that most books will stop talking about this dogma of biology. But actually, though, you need to go on and say, well, once you've built the protein, how do you make it into a mature protein? Like, what are you supposed to do now? And how do you bend it into what it's supposed to be? So in other words, you can say that I have a bunch of wood, but how do I make a table out of this wood? And so that's really where this is all coming together. And so we talked about the different stages of protein, where you have the primary structure, which is just this, sequence. Then you have the secondary structure, which is like the hydrogen bond structure, where it's like you have very basic bonding in your across your amino acids. But then you have the tertiary structure, where you have the one protein as it's kind of folded into this knot, this blob, and that's where it's beginning to take on it's beginning to be able to take on its duties. But its duties, most proteins will have its duties. A duty would be for a protein to interact with other proteins. So that's where the quaternary structure is, where one protein has to be able to connect with another protein. Now, in case it hasn't dawned on you, if a protein doesn't connect with another protein, imagine somebody gives you a box of Legos and none of the pieces stick together. None of them do. None of them have the studs on top. None of them have the special places on the bottom. They're basically just a bunch of like little plastic square blocks where they just don't stick together. Well, you can see that you can't build a house, you can't build a car, you can't build the little figurines, you can't do anything with that Lego because none of the pieces stick together. And so if you have some kind of problem in your tertiary structure in protein, where the protein is built in such a way that, or it misfolds in such a way that it actually cannot stick together, or it cannot bond or interact with other proteins, well, then guess what? You have some really awful, disorder on the horizon, because these proteins have to come together. They have to be able to work together in many cases. Anyway, there are some good videos here that you can look at. I hope that you did spend some time looking at these videos. I think that they're actually quite good. I watched them all. I can tell you that they're actually quite cool. But we spent some time actually talking about let me move on forward. Okay, we spent some time talking about some tools here. So here we are talking about the reasons for wanting to know about what proteins look like. But how do we actually find out what proteins look like? What kinds of tools are there? How do we find out where the sheets are, which look like these big kind of, corkscrew type structures? The coils, which are smaller corkscrews. The sheets, which are like these flat pieces of bendy, not turning, but bendy material. Or the turns, which are like these thin pieces of cord that exist between, that hold these structures together. Those are what they call the turns, like the twists. They usually hold things together, but there are specific pieces of your protein. And so the big question that we're asking is, okay, I get it. I see how there's this stuff in protein, but how do I actually find out where this stuff is? I mean, if I have a piece of protein, how can I find out what it looks like in some three-dimensional sense? And so one of the tools that we looked at first was this thing called Garnier, which is actually quite interesting. It's a cool little program. It uses an algorithm. It's a standalone program, but it uses like an algorithm, but that basically uses physiochemical type properties to say, when I find an M followed by a Q, like I'm looking at here, for instance, if I find an M followed by a Q, then I know that that's typically a helix. Not always, I guess, but typically it would be, because of the way that this, the amino acids would want to fold. When I find an I followed by an F and a V and a K, when I find those particular amino acids together, they have physiochemical properties which would inspire sheets to be made. And so it's going through the entire sequence doing exactly this. It's looking at specific types of protein, and it's saying, well, when I see this kind of conformation, when I see these kinds of amino acids together, that typically has this kind of structure based upon their physiochemical properties. And while that's good, it seems to work out quite nicely. In the end, what you get is, if you put your piece of code in like this, you just get this like long, in fact, this is the result you get from the bottom here, but you get a piece of output that looks like this, where you're just kind of following through the entire sequence. And in your mind's eye, you have to start putting together the ideas about, how this thing looks. But you don't actually get this three-dimensional blob that we've been talking about before. It's like you can't really envision the colors and the sheets and all that stuff together. But you do see, for instance, where, if you're studying, for instance, the boundaries, this is a study that I did, in fact, it's one of my papers in graduate school, but I was looking at the boundaries, for instance, between the turns and the sheets. What kinds of physiochemical properties do you expect to find right here, for instance? What happens at that border? And is that border there for a reason, or is it arbitrary? So in other words, we were playing with the idea that, for instance, if you have specific types of structures that are in your DNA, which are like magnets for bad things to happen. Basically, if you have these structures in your DNA, they attract bad reactions with amino acids, which could result in like an unraveling of your protein. Why do we have so many of those particular, I guess I'll call them bad words, but why do we have so many bad motifs or bad words in our proteins? And in our research, we found out that you had to have those bad words. You had to have those particular words, which would attract, undesirable interactions with other proteins, because that's how you would make the difference between certain types of turns and sheets. For instance, if you wanted to go from a turn to a sheet, you had to have a specific type of word there, or a specific type of amino acid with a physiochemical property to make that transition possible. But at the same time, if you had that specific word there, then that word would, or that particular, I guess I'll call it an island here, or that motif, could also work to attract other types of proteins which would unravel your first protein. And so in other words, you have to, if you're designing proteins here, you're having to decide whether, is it a good thing to have this word here? I need this word here to help me with my own protein structure, but if I have it, then I have problems with other proteins. And so what do I do? What do I do? And so that was the whole nature of the paper. I can share that with you I can share that paper with you if you're interested. But it was actually quite a lot of fun. And we used Garnier for this whole thing. And the funny thing about Garnier is that it comes out in 1978, which is a long time ago. That was an algorithm from a long, long time ago in terms of bioinformatics. But it still seems to work. Now, there are other places where you can get information about proteins, and this is where we're kind of starting some new material, and that is Uniprot. Uniprot is actually quite exciting. We've actually spent some time looking at Uniprot already, so I won't spend too much time here. I'm going to kind of move on. But I do invite you, though, to have a look at Uniprot. And what Uniprot will do is it gives you seemingly all available information about a protein. So, for instance, if you are working with a protein and you want to know about, like, for instance, its function, or maybe how it interacts with other proteins, or you want to know about what papers exist about that protein, or seemingly all information that you could possibly want to know about a protein, well, I guess not all information, but all information that has been discovered about that particular protein can usually be found at Uniprot. And so, in fact, I'll just go there just to see what the website looks like. But if you go there, you'll see that, for instance, you have this. You can type in seemingly any protein you want, and the protein I was working with was Cert I. That was one of the proteins I was looking at, which is one of those big proteins that seems to do a lot of work in many different arenas. But for instance, you can just click on these proteins here, and you have all this information about. This is about the function here. I mean, look at how much information there's about the function. This is a regulatory protein. It's like if you're making bread, you have to have yeast, right? It's like one of those common things. You just have to have yeast when you're making bread. If you're making proteins, if you're managing proteins, you tend to need this type of protein here to help you manage other proteins. And so it's kind of like your center protein to the whole function of any other protein. So you have to have these things. And so they are regulatory types of, they have regulatory types of functions here where they kind of control other proteins. So you can see that the functions are extensive. There's a bunch of things that this protein does. It seems like it does everything. It's involved in liver and muscle metabolism. So for instance, every time you are, if you're going to the gym and you're exercising and somebody says to you, hey, why don't you just like skip the gym and come and get a cup of coffee with me? And you'll say, no. I have to work on my Cert I protein today. I have to give my Cert I protein some, I have to put that to work. And so really you'll be, as you're building your muscles here, your Cert I protein is going to be kind of like coming together with your muscular proteins and making them, do the jumps and playing the basketball and doing all the things that you're trying to do in the gym. It'll help those things to help those muscles to perform. So anyway, there's a bunch of stuff here. And so what I'm trying to say though is you have all this information about these different things. What I was looking at when I was working with my proteins, where I was looking at this area down here, this PTM, which stands for post-translational modification. And that is the area where you're taking a piece of protein, like the sequence, and you are folding it into a specific type of function, or a specific type of form rather, so that it has a function. Form and function are intimately related. And in my area, though, I was looking to see that if you have a piece of protein which is folded in a particular way, could that folded protein be kind of unfolded slightly and then refolded in a different way so that it could compete with maybe environmental conditions. If some kind of, let's say that the heat goes up, and the function of the protein begins to feel some kind of toll. Maybe proteins don't like to function in hot areas, and then you have the heat going up for that particular protein. The environment where the protein is gets hotter and hotter. You might find that protein is unable to continue its duties. It's just unable to work. However, you'll find that in post-translational modification, that protein will actually, it's basically reconfigured in some way. It's undone slightly it's untangled in a certain way, and then a new knot is made in that area, which allows the protein to continue its function, even though the temperature has just moved up. And so they call that the post-translational processing, where you're able to take a protein and kind of repurpose it for a different type of environmental stress. And when that stress goes away, then it's repurposed again back to how its original state was. I mean, imagine how that would work for us as people if we found that we were just unable to work or unable to live, for instance, when the temperature became freezing. It could happen. When you go outside, it does get cold, your face gets cold, your hands get cold. But actually, though, you don't die because it gets so cold. And when you go outside in the summer heat, if it's like 100 degrees outside, again, you don't die in the summer heat, because your proteins are able to reconfigure themselves in certain ways to allow them to continue functioning, even though they're cold or even though they're really, really hot. And so that happens all the time. And when this discovery was made that proteins can actually kind of repurpose themselves to be able to continue their functions in spite of environmental shocks, it was a monumental, it was a breakthrough, a complete breakthrough. It's kind of like saying that discovering that your computer can actually work quite well underwater. I mean, we all know that if you take your computer and dump it underwater, it's not going to work. It'll break. It will just break. And so you have these proteins now, which I would argue are as complicated as a computer, but now they're able to function seemingly anywhere. Within reason. Obviously there are some extents where they can't function. If they're on fire, for instance, they can't work anymore. But what I'm trying to say though is in this particular thing here, all these references here, the amino acid modifications, these are all sites in the amino acid which are acting as like light switches, which means that if you can somehow switch on, I'll just click on one of these things here, if you can somehow get to this, if I can get to one of these things here we are, phosphorserine. If we can go ahead and click on one of these things here, you'll find that there's one base which is glowing yellow here. This is the protein sequence. This S, for instance, serine, I think it is. If you were able to somehow interact with this one S chemically, then you can suddenly reconfigure this protein. It's like that's a light switch in this protein. If I can push that S button by adding a methyl group or something else to it, then I can change how this protein folds, and I can change the function of this protein. So that is actually a very important piece of knowledge, in that, for instance, imagine there's one piece of protein here that's not functioning correctly because it's been misfolded. What if, for instance, you were able to turn off the bad folding part and kind of disable it in some way? Well, you could do that perhaps by working with these post-translational modification sites. And so this is kind of like you're hacking a piece of protein. And so it's kind of like, I mean, you're hacking a piece of code. It's like here we are in computer science hacking software. But if you're in the lab, in like the bioinformatics lab, you could actually hack a piece of protein code and make your protein start having different kinds of properties. Anyway, I think you get the idea. There's a bunch of other stuff in each of these, in this whole database. Seemingly all the information that you'd ever want to have about your protein is available here. This is actually something that's quite interesting here. These are your gene ontologies. Gene ontologies, I haven't mentioned the word ontology in class, but I'll tell you here that an ontology is it's like an appellation or a calling. How do I call this particular protein? So I may call this a binding factor. I may call it that in my research, but actually that's not all it does. I might say it's the binding factor, but actually somebody else might be working with this gene and they might find out that actually it's an enzyme binding factor, or they might call it a histone binding factor here, or maybe it's a nuclear hormone receptor binding. What I'm trying to say is that the names that are attached to each of these genes They're all different, but the gene is still the same. And so we have these gene ontologies, or these lists of words that we use to describe genes, so that if I'm working on a gene and you're working on a gene, you can look up what I call my gene, and I can look up what you call your gene, and we can actually find out that it's the same gene that we're working on. And so it just helps research to happen. Before they were using these gene ontologies, it's still not perfect today, by the way, though. It's still not a perfect system. But before they were using these gene ontologies, you'd find that many groups were working on genes, and they called, one person would call it, gene X, another one would call it gene Y. And then somebody who discovers the same gene, they would call it gene Z, but they would not know that it's already been discovered and people are already working on it. And so you'd have, maybe you might have 3 different research projects which are all working in parallel, completely unaware of each other. And so if you have these gene ontologies, at least now there's a chance that you'll be able to find out that your gene that you're working on is actually the same thing that somebody else is working on. It's still not a perfect system, and actually one of my research areas is to bridge that gap, to allow different types of groups in bioinformatics to discover each other, and you can discover another group based not on the actual names that you use for your proteins, but the actual ideas, concepts, and knowledge that you're using to work with those proteins, or those whatever you're working with in your lab. Anyway, I can discuss that later on if you want. The next thing I wanted to talk about, the next big tool, is something called predict protein, which is actually something, it's actually quite exciting. This is a tool which looks a lot like BLAST, but what it does is it gives us a little bit more information about how these proteins actually come together structurally. So in other words, we have this tool over here, which is Garnier, which is sufficient, and you can run this and have your results in seemingly seconds. Whereas if you run this, your results might be a little bit more factual, maybe a little bit more checked by, maybe they're compared with real results you'd find in nature. And so you're expecting a higher quality of results, maybe a more accurate type of result. The only problem, though, with this is that it could take you an hour to get your result, which is actually sometimes, that's actually a long time to wait. I mean, would you wait for an hour for a website to open? I mean, it's hard enough to even wait for 3 seconds. If a website doesn't open in 3 seconds, I feel like I'm gone. That's it. I'm not going back to this website. And so here, with this predict protein, you do have a more kind of, there's more analysis going into your protein to figure out what it looks like in terms of its structure, but it does take longer to do. And so you have all this you have all these different types of annotations also that it gives you. So let's go ahead and just look at this. And so, for instance, I call this these are slower solutions. And slower solutions are exactly what they sound like. You get a good solution. I mean, it seems to be a workable solution here. It seems like a workable thing. But it does take much more time to get. This is, by the way, if you click on this graphic, this is a link. And so when I click on this graphic, I get something like this. And so you can just go ahead and fill in seemingly any protein sample that you want. And I'm just clicking on the example down at the bottom here. But when you click on the example and you click on the predict protein, it takes this protein. This is a protein sequence, and then it does a whole bunch of work. This is called the Smart7 Human. This is some kind of a protein from human. And if you go down to the bottom here, it gives you some ideas about different kinds of annotations that it has found. And so I like the analysis on this thing here, though. This actually doesn't take much time at all to run, because this is an example sequence, and so they built this into the software as a macro. But if you give it your own sequence, it could take more time than this. But for instance, you do have some information about the types of annotations that exist in that protein. What do we have here? These are this is an alignment here, so it's actually looking to see what other sequences have similar types of alignments to the one that we're looking at here. And you can zoom in and look at things, which is actually quite cool. I think that, I'll state that. But one thing that's actually quite interesting, though, is this amino acid composition here. And you're probably wondering, why would anyone care about the amino acid composition? Well, as we discussed last time, supposing, for instance, I know that that human DNA or human proteins tend to focus on specific types of building blocks. Maybe there's more of these kinds of building blocks in human DNA or human protein than other types of protein. And so if you give me a piece of protein and I find a bunch of those building blocks in here, like a bunch of, say, Gs, I might say, well, that's funny. I know that humans have a lot of G in their protein, or in this particular protein. I know that this particular protein has a lot of G's in it when it comes from human. However, when I find similar types of proteins that have similar functions, I'm not exactly sure what protein this is. If I find that that protein has like maybe a bunch of serine or a bunch of leucine in there, then I might suggest that perhaps this, I could kind of guess something about the origins. Perhaps I could say perhaps this isn't a human origin type of protein here, but maybe it comes from a fish or something. But you do have like these basic statistics here, and while sometimes that information isn't always handy, it certainly is interesting though to find out like what are the basic compositions of this protein. And you might find, for instance, that there's a study available to you just by looking at the kinds of compositions that go inside these proteins. You may find, for instance, that certain types of proteins require specific physiochemical properties to function, and you'll find that in your amino acid composition, the most popular or the most commonly found amino acids are those amino acids that have those physiochemical properties. And so there you are. So you might say, well, I can now start grouping my proteins together according to types of abundant physiochemical properties. So you do have some really cool stuff that exists in here, and you have, of course, the sequence length. You can have different ways of looking at your sequence. I'll just show you the different ways of looking. We're used to looking at our sequences like this in FASTA, where the first line, this should be the title. The first line, this 426 BP. They don't have a title there. They just have 426 BP. But in terms of a format, that's still a perfectly good. That's still a perfectly good title, and the actual the code for the sequence follows on the second line. But you have other ways of looking at this code, and so for instance, you have a giant grid that looks like this, which allows you to kind of go through piece by piece by piece, and this allows you actually to find specific reaction sites. So for instance, if I want to look at like reaction site number 5, oops, it's right here. Or if I can point out of things like here, I can say this is the last reaction P, or this is the last amino acid, and this is the first amino acid. Note again that this amino acid begins with M. Most proteins begin with M. And these other ones here, these are just different types of formats you'd use for different kinds of software that would help you to work with them. So you get the idea that you have a whole bunch of information here about these proteins which exist. But still, though, you're looking at when you run this tool, though, sometimes it takes a long time to give you some ideas. I think we can just click on some stuff here. And so here we have like secondary and some secondary structures. Can I click on this? Here we go. So this is actually where we are today, though. We're looking at the function or the structure rather of different types of code or of different kinds of protein. And what we're seeing here is that like in this analysis, you're finding that this first part here is a helix from 5 to 15. This is other, which means it may not be relevant to many of the studies where this protein was found. If I click on here, it's another helix, another helix. Over here, this is a strand, which is like your cord, I guess. Strand, and so you can kind of zoom in and see what your code or what your protein is going to look like. But again, this is very similar to Garnier, which is like this system here, where you're just looking at the protein going line by line by line and getting this idea of what the code looks like as you're at different areas. But it does provide some good information, though. I mean, you can see, for instance, what your sheets and your chords and what those other things are, where they are, and maybe that would give you some idea about how they're working in the sequence. Like, how do your sheets actually allow this protein to interact with other proteins? So yeah, let's just say I know that, for instance, for my sheets to work, I know that they have to be in. Or for this protein to work with another protein, I know that I have to have a sheet at exactly. This is another goo, by the way. But I have to have that. Here we are. Let's do this helix here. But I know that, for instance, for this protein to work, I know that I have to have my helices at exactly this position. And if they're not at this position, then I know that this protein is not going to be able to interact with a specific molecule of protein that it should be. And so if I don't see those helices, or if I don't see those coils or those helices at that position, then I know that something's wrong. And so you have this way of like looking at your results. In fact, how do I find that result? I think if I click on display my oops, not that. Somewhere I was clicking around and I found it. I don't see it here, but it is in here someplace. And so you can click around and find all the You can find all the different ways of looking at your sequence. There's a bunch of different ways. Anyway, so that's cool. But the next thing, though, is even more cool. This is called the Protein Data Bank. And remember how I said that it takes like a long time to figure out what the conformation of a protein is? Believe it or not, actually, in order to figure out this particular function here, or to figure out what this protein looks like in space, this could take you all morning to run this analysis on a fast computer. I mean, if you run, for instance, going back here, if you run this this tool here, protein folding, Solar Solutions, if you actually enter your own solution into that, your own protein into this, and hit run, and you wait to see how long it takes you, you might find it takes you up to 5 minutes for a sequence of protein, which isn't that long. But here, Protein Data Bank offers you huge proteins here, and you can work with them right now because they have all been sequenced. They've all been worked on. They've all been processed. So that means that around the clock, the good folks at the Protein Data Bank, or PDB, were running a supercomputer, and they still do this, but they're basically taking in sequences of protein, and then they determine what those proteins would look like as they are folded into their conformation. And so that then allows you to choose the proteins that they've worked on, and I don't know how they choose them. I think, but they have a fairly sizable, seemingly all the proteins that you'd ever really want to know about. Well, for some of your research, are there. It's like, and I think they're always adding to this database, and they always have the supercomputers that are running in the background that are adding more and more folded proteins to the mix. But really what it does is it offers you a three-dimensional view of your molecules. And so I can show you. Oh, by the way, if you wanted to run this locally on your machine, this isn't actually done anymore. It's kind of, old hat when you think about like running a virtual machine, like a Java virtual machine on your own machine. But you can still download a tool called JMOL, which allows you to download files from the protein database, which are really just like files that contain information about where exactly each atom is actually located in space. And then it puts all that information together, and then it allows you to create this viewer, or you can actually, it allows you to use this viewer here to visualize where these things are. So there's that. But if you don't want to use that, then that's fine. I mean, I can completely understand not wanting to install any unusual software on your machine. You can still go to their website and you can run all that stuff there. And so they provide their own interactive environment where you can check out what a protein looks like. So it does take a long time to fold proteins, and so this website actually has folded them already for you, and you can just come in and check them out and see what they look like. So I'm going to go ahead and click on this. This is a link, hopefully. In fact, I think this, hang on, in doing that, I've just lost I lose my slides every time I do that. There we go. So here we are at the website. And what I'm going to do is I'm going to search, or I'll just type in a search here. I'll just do Cert I again, just because it's my old favorite molecule. And you'll see that you have a whole bunch of different proteins that they have found, which are all categorized as Cert I. These are all those governors, if you will, of protein interaction. And you can see that there's a picture here. I'm just going to go ahead and click on the picture just to get there first, just to see what that is. But this is the record of the protein. I can get an idea about, for instance, other types of experiments which have been used in that protein to figure out, how that protein responds to specific types of environmental factors. This is actually quite interesting. I really enjoy this part here where you have the literature so clearly available. That means that all these researchers here are the ones who bring us the information on this page about Cert I, Cert II, and Cert III. They're doing some research on these three different proteins. They're basically three sisters who manage proteins in very much the same way. I guess three queens of proteins, if you want to put it in that term. But these are all proteins that govern other proteins. And you can find out more about this in this paper, which is actually quite nice. But what I really find interesting, though, is if I go ahead and click on this 3D structure, like I'm right down here, if I click on that, hopefully I can get this thing to come up. There we go. It might melt my machine because it uses up some computational power. But I have this thing that I can actually click on now, and I can see what the protein looks like. And I can zoom out, and I can zoom in. And I can see all kinds of some of the different pieces of information that have come together from folding, and I can see how they actually are in 3D space. This is actually quite important, though, because imagine, though, that, for instance, I am making a protein, or I'm making some kind of protein which is going to interact with this protein, and it's supposed to have some kind of therapeutic result. In other words, I want a protein that might stick if I can get in here. right here. I want something that sticks right there. That my protein, I found out from my analysis, that my protein has to stick to this piece right here. It just must. If it doesn't stick there, then that means that I'm not going to, my therapy is not going to work. For whatever biochemical reason it is, it must stick there. So then what does that mean for me? Well, I can go ahead and I might want to look around and, oops, I can kind of, I'm going to zoom out here and I can see like what the chemicals are that are inside, like what they kind of, what the biochemical or biochemistry interactions are around that area. But also though that I can zoom out and I can see, oops, so I can just get back to where I was before. I can see, for instance, how much space do I have for my new molecule to get in there? It's kind of like if you're parking a giant truck in a small driveway, you have to kind of like know, well, how much space do I have? Can I even get this truck in the driveway? Can I even get this protein to bond to this particular place in this protein? And so you have all these actual physical types of parameters that you have to abide by. And so this will allow you to model that information. You have other information here that you can look at too. For instance, you have components. You can kind of get an idea about what the different kinds of molecules are here. This is these are the polymers, and so if I click on this I can see the polymers. I can see what the ligands look like, and so I can kind of like, I'll just remove all these things here. And so if I have the ligand open, then I can just like study like specific parts of my protein. Where are the waters? These are my waters right here. You can kind of get an idea about what they look like. You can all see these. And for instance, what are my ions looking like? There's only one ion in this thing, which is right here. And where does that ion fit together with everybody else? Right here. And so by clicking around, you can actually learn very visually about not just about the protein itself, but how that protein can actually be used in some kind of how it would function, but also how you can create a therapy from it. And so that's really all I have today. I do recommend though that you spend some time looking at like certain types of proteins. Like for instance, this one here, the RCSB. If you go back over here and you do another search for that, let's see. Let's get back over here. But if I do a search for that here, I think you'll find that this is the I think it's the spike from the coronavirus. Is that right? It's one of those things. Anyway, I think it has something to do with that. And so you can get an idea about what how that protein would behave. And so you have all this information here. But also though, you can take this protein and you can compare it with other types of proteins that have similar functions. And after a while, you'll start seeing how these coils and these helices and these cords come together and make the protein function. I mean, if you're a biochemist, this is a very important tool for you to have. Anyway, that's all I have. That's basically it. Please do spend some time looking at these tools, though. I think you might find that they're actually kind of cool, and certainly though, if you're just playing with this PDB tool, you might actually find some really interesting proteins that just by looking at these things, you suddenly start realizing how they would function. And you just think, oh wait, that's probably how it would have to function. I know this protein docks with something else. It must function in that way. It must be this must be the method that allows it to dock with another protein.